My name's Brian Dickie and I'm Chief Scientist at the Motor Neuron Disease Association. So motor neuron disease is a complex disease, a multifactorial disease. It involves genetic predisposition, environmental factors as well, but all against an aging brain, an aging body that's less able to cope with any stresses that are occurring to the nerve cells. So we see the genetics as a route in to understanding all forms of the disease, because in some families where there's a high genetic risk, they are primed to develop the disease later in life. It doesn't mean they will develop the disease, but it does mean that there is potentially a route in, and we can therefore identify the pathways that are involved in their particular form of the disease by taking the knowledge into the laboratory, and through different inherited forms of the disease start to cross-reference. So it's a little bit like a river flowing out to the sea. A river has many, many different sources that form tributaries that come together into one final common pathway. And so MND, we know, will have many, many different causes. So we can start to map that out through looking at the inherited forms of the disease, work out through that what are the pivotal cellular processes that are going wrong within a motor neuron. And if we can identify those, that's where the drug industry becomes very interested, because that's what they call a drug target. Something is going wrong, we can come in and try and change that, alleviate it, hopefully protect the nerve cells and slow down the disease progression, not just in the inherited forms, but potentially in all forms of the disease. So the MND Association is investing in a number of areas of research. First is genetic research to try and identify the remaining genes that are involved in inherited MND, but also maybe the more subtle genes that play an underpinning role on sporadic motor neuron disease as well. Then taking the knowledge into the lab and trying to mimic in the laboratory, in a cell in a dish, in a fruit fly, perhaps in a mouse model, the events that are happening in the person living with the disease. Another key area is what we call biomarkers, which is, can we identify changes, for example, in a blood sample or through a brain scan, how the very earliest effects of the disease are manifesting. Now, this could be years, maybe even decades before the first physical symptoms of the disease appear. And in order to do that, we actually need to study people who carry an at-risk gene. It doesn't mean they will develop the disease, but it means that they are primed and therefore more likely that we will see something happening at the very early stages of the disease. And that's absolutely crucial for two reasons. The first is that we're not diagnosing this disease quickly enough at the moment. And common sense dictates that the sooner we diagnose a disease, the sooner we treat it, the more effective these treatments might be. So we're missing a crucial therapeutic window of opportunity. The other reason is that if we can understand the earliest stages of the disease long before the physical symptoms manifest, then possibly there's an opportunity to be able to prevent the disease from ever occurring in the first place. And that I see would be an ultimate goal. Prevention is better than cure, as we say. Events like the FaTHoM meeting today are just so important, because for all the science that's being done, we always have to ensure that it comes back to the human condition. We're not here to cure fruit flies or cells in the dish. We're here to treat and cure people. And we need to be working with people with MND and their families, particularly those with the at-risk genetic forms of the disease, in order to achieve that aim.